Hello, this is Professor Miller, and I'm here with a quick lab introduction to Chapter 13, where we'll talk about Hardy-Weinberg equilibrium and these two equations, which you really need to understand how to use, and then also introducing Chapter 20.2, where we look at the plant-like protists. So let's start off with Chapter 13, thinking about Hardy-Weinberg equilibrium. Some review definitions that you should be comfortable with. First off, a gene. A gene is a sequence of DNA that usually codes for a protein of some sort. You would need to think of genes as generic categories, something like hair color, right? That's just a big generic category. Genes over time have mutated, and now we have multiple versions of genes. Those multiple versions of genes are called alleles. So if the gene is to generic hair category, something like hair color, then the allele is something like brown hair or blonde hair or black hair. Every individual is going to receive two alleles for any gene that they have. If those two alleles are the same, we say that that individual is homozygous, and that's represented usually with either two uppercase letters or two lowercase letters. If those alleles, however, are different, those individuals are called heterozygous, and they have both a dominant and a recessive allele. All of this is good because what it means is in a population, we end up with a population that has variation. Variation is good in populations. We want that. It allows natural selection to work upon populations. Some other terms that we just want to make sure that we're comfortable with. Evolution is the change in allele frequencies over time. It may happen at a very small level, or as we saw last week, it may happen at the species level or higher. Natural selection is the mechanism. It's the thing that does the evolution for us. And through the process of natural selection, we end up with adaptations. So here's a great definition of evolution. Evolution is not always creating a new species. It's not having this brand new thing that we've never seen before. Evolution at its basic level is any change in allele frequencies in a population over time. Note that this is happening at the population level. Individuals will never ever evolve. If you're born, you are going to die that same individual. You don't evolve, but your population may evolve, so that group of individuals may evolve. So it's important to remember what these terms mean. A population is a group of organisms, all of the same species. So for instance, it might be all of the bunny rabbits that live in McKinney, or it might be all of the oak trees that live in Yellowstone. Gene pool is when we take that population and we look at all of the genes, whether individuals are homozygous or heterozygous for a particular trait. And then the allele frequency is a number we can calculate telling us the frequency of those dominant and recessive alleles. So what evolution becomes now is if that allele frequency is changing in a population, we say that that population is evolving. Whether or not we're getting speciation really doesn't matter. As we think about microevolution, it's just that change in allele frequencies over time. So let's go ahead and sort of walk through some of these terms and see if we can make sense of them. If we want to figure out if a population is evolving, we have to be able to figure out what the allele frequencies are. So I'm going to take a hypothetical population, and instead of 10 moths, let's go ahead and just do 5 moths. It'll be a little bit faster. Some of these moths are black, some of them are blue. You can see their different potential genotypes over here. We're just going to choose a couple randomly. So I have one that is Big B, Big B. There's a nice black one. I've got another one that's Big B, Little B. That's another black one. I've got another one that's Big B, Little B. And I also have some that are blue. I've got 2 blue ones, Little B, and then another one that's Little B, Little B. So here's my population. I've got 3 black moths, and then I have 2 blue moths. So this is just our pretend population. What we can do in this population is we can count up the alleles. Again, the alleles are those specific versions of the gene. In this case, we're representing it with a big letter B or a lowercase letter b. So if I count up all of the big B's that I have, it turns out that I have 1, 2, 3, 4 big B's. I'll just keep track of that. We had 4 uppercase or letter B's. And then if I count up all of my little letter B's that I had, we'll keep track of this as well. We've got 1, 2, 3, 4, 5, 6. So let's keep track of that as we go to this next page. We've got our hypothetical population. We've got the 5 moss, the 3 black, the 2 blue, and we figured out that we have 4 total of the big B allele and 6 total of the little B allele. So I'm just going to write that down again so we don't forget it. The big B allele, we had 4, and for the little B allele, we had 6. What we can do is we can take these values and we can figure out the frequency of each allele. You'll note in our sample that we had 10 total alleles, not 10 individuals, but 10 total alleles. So we can take these values and we can change them into frequencies. So the frequency of big B becomes 0.4 and the frequency of little b becomes 0.6. By convention, the frequency of the dominant allele, represented by the uppercase letter, we refer to as P, and the frequency of the recessive allele we refer to as Q. The following proves to be true in populations. The frequency of the dominant allele, P, plus the frequency of the recessive allele, Q, will always equal 1. And you can see that works here with the example that we just went through. 0.4 plus 0.6 equals 1. This is a really useful equation to know. You should be comfortable with it. You should be able to work with it, especially on a quiz or on a practical exam. But please remember that this represents the allele frequency. That's the trick here. This is just telling us the frequency of these dominant and recessive alleles. Hardy and Weinberg, these two gentlemen we see here on the slide, described what would happen when populations found themselves in genetic equilibrium. This is a way of saying that populations are not having evolution. In order for Hardy-Weinberg equilibrium to hold true, we have to meet 5 conditions. You should memorize and know these 5 conditions. They include the following. Number 1, in order for a population to be non-evolving, there has to be no mutation. Number 2, there can't be any gene flow. People can't be moving in, people can't be leaving. Number 3, there can't be any selection. So there can't be any natural selection. We can't be selecting for the tallest or killing off the red ones. Number 4, mating has to be completely random. There can't be any choice. Any female can mate with any male. And number 5, Hardy-Weinberg equilibrium has to have a very large population in order to function. These 5 conditions very rarely are going to exist in the natural world, but it's a nice hypothetical so that we can sort of see how this might happen. With the Hardy-Weinberg equilibrium, they developed this equation that describes populations that, again, you should memorize and you should know and understand how to use. P squared plus 2pq plus q squared equals 1. This quadratic equation is doing the following. The value p squared represents the number of individuals in a population that will be homozygous dominant. Note that it's the frequency of individuals who will be homozygous dominant. Frequencies are always going to be numbers between 0 and 1. So it's never going to be something like 66. P squared will be things between 0 and 1, so 0.6 or 0.34. Those will be the types of values you'll see. The middle term, P2PQ, this represents the frequency of heterozygous individuals. So this might be something like 0.54 or 0.16. Q squared, our final term, represents the frequency of homozygous recessive individuals in the population. Again, this is going to be a frequency, so a number between 0 and 1. The important thing to remember about this particular equation is that this is talking about the frequency of individuals. This is different than the previous equation, which was looking at allele frequency. So with these two equations, we can start practicing Hardy-Weinberg questions. As you answer these types of story problems, here are some tips for how to answer a Hardy-Weinberg question. First, you want to always start off with what you know. As you read the problem, you will be given some piece of information. You need to figure out before you even try to answer the question, were you given p or p squared? What is it that you already know? Second, before you try to start answering the question, figure out what is being asked for. Are we asking for you to figure out p, or are we asking you to figure out q squared? And then lastly, be very, very careful. You need to watch out to look for the difference between allele frequencies. If it's that, we're going to use the P's and Q's. Or if we're talking about individual genotypic frequencies, then we're going to use our quadratic equation, p squared plus 2pq plus q squared, or the number of organisms in a population. Let's talk about this bottom one for a little bit, the number of organisms in a population. If we're asked to calculate the number of organisms in a population, for instance, maybe there's 700 individuals in the population and you're asked to figure out how many are homozygous dominant, you are going to need to do the following. There are 700 individuals in the population, and you're trying to figure out the number that is homozygous dominant, so the p squared value. You're going to have to multiply those two numbers together to figure out what is this number of individuals in the population that have that particular trait that you're looking for. We're going to go through two examples to try to practice this right now. So here's our first practice problem. We have a population of 500, 245 are homozygous dominant, 210 are heterozygous, and 45 are homozygous recessive. The first thing that it asks is for us to calculate the frequency of the dominant A allele. Okay, so when we're trying to figure out frequency of the dominant allele, we just need to count up the total number of letter A's and divide them by the total amount of alleles. Sort of like we did with our moths, just this is a lot more. So in this case, when I'm trying to figure out the frequency of the big A allele, I've got 245 plus 245 plus 210. The reason that I did 245 twice is because you'll notice that of those 245, they had two big A alleles. So I add all this up together, and this equals 700. As I look at these individuals, I now need to divide it by the total number of alleles. The total number of alleles can be figured out by taking the number of individuals and multiplying it by 2. So 245 plus 210 plus 445 equals 500 times 2 equals 1,000. There are 1,000 total alleles. So 700 divided by 1,000 equals 0.7. So to calculate the frequency of the big A allele, it equals 0.7, or in other words, P equals 0.7. To calculate the frequency of the little a allele, we now have two choices. The easiest way, I think, is to use our equation where if you know P, you can solve for Q. We know P is 0.7, and so in this case, we can just quickly figure out that Q must be 0.3. Or if you want to check your math, we can go and add it up just like we did in the previous situation. So let's go ahead and get ready to give ourselves some room to do this work. If we want to add up all of the individuals that have that recessive allele, we have 210 plus 45 plus 45, and if we add all of that up together, it equals 300. Sorry, that's getting pretty sloppy. 300 divided by my total of 1,000 gives me 0.3, which again is what we had figured out. So we've already answered the first two questions. We figured out what P was and we figured out what Q was. The last question asks us the following. The second generation has 800 individuals. The population is not evolving, so we can use these Hardy-Weinberg equilibrium equations. How many individuals will have the genotype big A, big A? So we need to stop for a minute and make sure we understand what is being asked. Note, they're asking about individuals, So we're going to want to use that quadratic formula. And note that I'm not asking about individual frequencies, but actual number of individuals. So here's how we would go about setting this up. To figure out the individuals who have genotype big A, what we're going to do first is use the quadratic equation p squared. P squared represents the frequency of individuals who are homozygous dominant. We will then need to take p squared and multiply it by our population size, which in this case is 800. So p squared in this case equals 0.7 squared times 800, and we pull out our calculators and do that math, and the answer is 392. So that was our first practice problem. Second practice problem. In this practice problem, you start off with the percentage of homozygous recessive individuals in a population is 36%. So again, before you read on, you need to stop and figure out what you've got going on. You've been given the percentage of homozygous recessive individuals. So that's not P or Q, those are individuals. Percentages are similar to frequencies. 36% is the same thing as saying 0.36 as a frequency. So the information that we've been given right here is the frequency of individuals who are homozygous recessive. Little a equals 0.36. Or in other words, this is Q squared. So again, we haven't tried to even answer any questions. We've just tried to figure out what was given to us. So by that first statement, we've been able to figure out that Q squared equals 0.36. All right, let's look at the questions. First question, what is the frequency of the little a genotype? Or in other words, what is Q squared? Oh, well, we already know that. That's what was given in the question. That's 0.36. All right, let's go to the next one. What is the frequency of the big A allele? In this question, we're specifically asking about an allele frequency, not an individual frequency. So we need to use the equation P plus Q. The only thing we know right now is Q squared equals 0.36, but from here we can work backwards. What we can do with this equation is we can take Q squared equals 0.36 and we can take the square root of it. From here we can figure out that Q equals 0.6. So I'm just taking the square root of everything. And then if we know that Q is 0.6, from there we can figure out that P, not B, P equals 0.4. So the question is asking what is the frequency of the big A allele, or in other words, it's asking what is P, and we went ahead and we figured that out. P equals 0.4. All right, third question. The population contains 500 individuals. How many will be heterozygous? Note again that we're not asking for frequencies here. We're asking for individuals that are a particular trait. So we're going to have to do the same thing that we did on the last practice problem. So we'll start off with our 500 individuals, and then we're going to multiply that by the frequency that will be heterozygous. In this case, that's 2 times P, which is 0.4, times Q, which is 0.6. If we go ahead and do this math, it turns out that the answer is 240 individuals will be heterozygous. You'll want to spend some time becoming comfortable with these Hardy-Weinberg type questions. There are more practice scenarios for you to work through on the minimum requirement sheet. When you start the lab for Unit 13, the first thing that you're going to do is a procedure where you are given 50 yellow beads and 50 red beads in the same beaker. What this is representing is the possible alleles in a population. So you have 100 alleles in this fake little gene pool. What you'll do is you'll reach your hand in, and without looking, you'll pull out 2 pop beads and you'll write down what their colors are. If it's a red bead, you're going to use big R. If it's a yellow bead, you're going to use little r. So you've got 3 different choices. Sometimes you'll pull out 2 reds. If that happens, then you'll write down big R, big R. If you pull out a red and a yellow bead, then you'll write down big R, little r. And if you pull out 2 yellow beads, you'll write down little r, little r. After you've pulled out the beads, you return them to the bucket, mix it up, pull out 2 more. Oh, they're red, red. Okay, big R, big R. You put the beads back, you pull it out. You do this 50 times just to represent 50 babies being born from the population. At this point, you'll then stop and you'll figure out in those babies, those 50 individuals that were just born, what are the allele frequencies. You're going to figure out P and Q. Once you figure out P and Q, they probably will have shifted a little bit. They're not going to be 0.5 anymore. They're going to have moved. You'll then change your cup to match the new allele frequencies. So when you started off, everything was 50-50. There were 50 red beads and there were 50 yellow beads. If after going through the first generation, everything has changed and now P is 0.4 and Q is 0.6, you will change your bucket so that you'll have 40 yellow beads and 60 red beads. You then do the exact same thing with that new bucket now. Pull out 2, write them down. Pull out 2, write them down. Pull out 2, write them down. And you'll just keep doing that over and over again. You'll repeat this for 3 generations. And then you'll plot the frequency of the dominant allele on this table. Those of you guys that are in the morning lab, this data has been provided for you. You can find table 13.9, the raw data for this experiment, in our Canvas course. You then go to, sorry, that is not correct. The raw data for procedure 1 is the data that fits into table 13.6. All right. Now moving on to procedure 2. In procedure 2, you're going to do the same thing, but in this case, we're going to imagine what's going on when there is some sort of selective force working at play. So in this case, we're selecting against yellows or for the reds. How do we know that this selection is going on? Well, the yellow is going to be lethal. So you're going to do the exact same thing, 50 yellows and 50 reds all in the same container. And then what you're going to do is you are going to pull out 2, write them down. Pull out 2, write it down. Pull out 2, write it down. Even if you pull out 2 yellows, 2 little r's, you're going to go ahead and write it down. Once you're done, stop, calculate P and Q. But this time when you're calculating P and Q, you cannot calculate this using those recessive homozygote individuals. They're dead, so you're going to need to calculate it differently. Follow the instructions that are there on page 176 and 177, and it'll walk you through this. You're going to do this for 3 generations, and you'll then graph these values as well. So on your chart, you're with a different color, maybe with the blue one that I've got here, you're going to show how your allele frequencies changed after they started off at 0.5, and then what they did in that first generation, second generation, and third generation. Again, my morning lab, you guys have the data required to complete this experiment. Table 13.9 is provided for you in Canvas. The last procedure is to look at what happens when small populations evolve. So in this case, what you're going to do is you are going to pull out 10 beads, 5 yellow, 5 red, and you hold them in your hand. And then you really quickly have a baby. And you're going to always replace those 2 beads, and you're going to pick for 5 individuals. So here we've got some sample data to look at. As soon as you do this, you need to stop and you need to calculate P and Q. This is quite easy because there are 10 total alleles. You can just count up the total number of big R's or little r's. So in this case, we have 1, 2, 3, 4, 5, 6 big R's. So the frequency of the dominant allele, or P, equals 0.6. And then we can count up the recessive alleles, 1, 2, 3, 4. And we can calculate the frequency of the recessive allele, or Q, which is 0.4. You're going to do this 6 times. You're not going to change up the population in between. You're just going to quickly pull 5, start over. Pull 5, start over. Again, you're always putting the babies back, and you're trying to just see what happens to these allele frequencies when we've got a small population as opposed to those larger populations in procedure 1 and 2. After completing this, we're going to then move forward to chapter 20.2 to look at the plant-like protists. In chapter 20.2, I'm flipping there myself. These plant-like protists are sometimes considered algae. This is a non-taxonomical name. It's a very sloppy name. It just is referring to those protists that are plant-like in size. You do require apron and gloves today. We are using live specimens, so please do be safe. We are looking at 5 different plant-like protists. For each of these, you should be able to identify the phyla name and then its name. For Volvox, Spirogyra, Polysiphonia, and Euglena, those are scientific names. Diatoms is a common name. For each of these, you should understand how they obtain their nutrients. All of these are photosynthetic because they are plant-like. And so, for all of them except one, for Volvox, Spirogyra, your diatoms, and Polysiphonia, their nutritional mode is going to be photosynthesis. They are autotrophs. The one that's sort of the outlier is this Euglena, this bottom one. Euglena is interesting because for its nutritional mode, it's a mixotroph. Euglena has the ability to photosynthesize, so it's a plant-like protist, but it also has a mouth and it can eat you. So it's also acting as a heterotroph. And so Euglena is the only organism that we're going to look at in lab that turns out to be a mixotroph. So that's something unique to remember about Euglena. The first one that you'll look at is Volvox. You should know that this belongs to phylum Chlorophyta. They're photosynthetic, and the unique thing about these is that they're colonial. You're going to look at a prepared slide, and you're also going to look at a wet mount. As you look at them, try to notice the individual units, which are those little dots. Those are individual protists. And then what happens is they form together to make these big colonies. So this entire structure is one big colony of Volvox. It can reproduce, and so you can see these smaller daughter colonies inside. Eventually what will happen is this whole ball will explode, and those smaller daughter colonies will go out. Procedure 2, you're going to look at an organism called Spirogyra. This belongs to phylum Chlorophyta. It's also a phototroph. It's able to photosynthesize. These reproduce sexually through a process called conjugation. So this is what we see happening in this top picture and on this particular poster. This is a way of sharing genetic information. The nice thing about Spirogyra, the way that you'll be able to identify it, is because it has this nice spiral chloroplast. And so you can see that spiral chloroplast. You're going to be looking at a slide of this, a poster, and a wet mount. In procedure 3, we're looking at the diatoms. With the diatoms, remember this is just a common name. There are lots and lots and lots of diatoms. So please make sure that you've looked at a variety of them so you're comfortable with their very different shapes. They belong to phylum Basilariophita. They are photosynthetic or phototrophs. The unique thing about the diatoms is that they have a silica cast or a test. So the shell on the outside has the silica, making them look sort of glass-like on the outside. And this also makes them really hard to decompose. So we get these silica tests preserved very well. You're going to look at slides, a wet mount, and there's also a poster of these. So here we've got the slide. You can see some of them look like folded over footballs. Some of them look like rulers. Here's some of the live ones. Here that's easier to see that they're photosynthetic. You still have that green at center. And then these orange things over here on this particular poster are also diatoms. For the 4th procedure, you're going to look at Polysiphonia. It belongs to phylum Rhodophyta. This is another photosynthetic protist. The red algae, or things that belong to phylum Rhodophyta, are red in color often. They are red because they contain a component called phycobilin. This is a pigment that gives them their red color. On this, you're going to look at the slide. You should be able to identify Polysiphonia from the slide. In addition, though, there are a bunch of preserved blocks that you should also look at. With the preserved blocks, you should be able to identify any red algae as belonging to phylum Rhodophyta. Procedure 5, we're going to look at Euglena. This belongs to phylum Euglenophyta. That's nice, sort of the name gives it away. This, again, our exciting thing here that we can talk about is that this is a mixotroph. These do have an eye spot, which is often hard to see, but that eye spot will form at one end and helps orient them towards the light so that they can then photosynthesize better. You are going to look at a slide of them. The slide is stained, so they're going to appear blue. It's hard to see their flagella in the slide, but they do have a flagella that they use for motility. You'll also get to try to look at a wet mount of these. They move very quickly. You may have to keep it at a lower magnification so that you can catch one. So those are the things that we're doing in chapter 13 and chapter 20.2. If you have any questions, as always, feel free to ask.